Hi, I'm Tom Lynch, President and Director of the Fred Hutch Cancer Research Center, and welcome to our virtual event, Vaccinating the World. Today we're going to hear about what it will take to develop and distribute vaccines for COVID-19. Before I introduce our two presenters, many of you may be new to the Fred Hutch and wondering why a Seattle Cancer Research Institute was able to pivot to be working on COVID-19. First, the Fred Hutch has long been an innovator in immunology and immunologic-based therapies for cancer. Bone marrow transplant started at the Fred Hutch. Using cell-based therapies to treat cancer was something that was pioneered at the Hutch as well. We have great expertise in understanding the immune system. Second, we have a 650-person vaccine and infectious disease division working on HIV, TB, influenza, and other viruses. You're going to hear a little bit more about that from Larry Corey a little bit later on. And third, we have experts in disease modeling, tracking, tracing, and public health to help track and understand the societal causes of cancer and infectious disease. As many of you know, infectious diseases and viruses are responsible for approximately about 30% of human cancers. So that connection between cancer and infectious diseases is very tight. So when COVID-19 hit, our researchers were able to apply their expertise to this new health crisis. I'd like to start by introducing our two speakers today, two presenters today. First is Dr. John Mascola. John is the director of the Dale and Betty Bumpers Vaccine Research Center at the National Institute of Allergy and Infectious Diseases. John recently co-authored an article in Science with Dr. Anthony Fauci and Larry Corey, as well as Dr. Francis Collins, talking about the importance of a collaboration between academia, government, and industry to come up with good options for vaccines. We'll also be joined today by Dr. Larry Corey. Larry is a professor of Fred Hutch's Vaccine and Infectious Disease Division. He was the fourth president of the Fred Hutch, president and director of the Fred Hutch, and he's also been the principal investigator of the HIV Vaccine Trials Network, which is based at the Fred Hutch since its inception in 1999. So I'm just going to start off by quoting a line that's in the article that appeared in Science that John and Larry wrote. And I think it's really an incredibly important line. It's one of the last lines of the article, and it says, "To return to a semblance of our previous normality, the development of a SARS-CoV-2 vaccine is an absolute necessity." John, tell us why you wrote that.
Well, thank you, Tom. I really do appreciate the invitation to participate in the event today. So, I'll start by giving you a little bit of perspective about how we think of vaccines in the vaccine community. So how do we think about developing a COVID vaccine? And I'll say up front that there's a lot of optimism in our community that a vaccine should be possible, but we are very focused on the fact that that has to be proven in clinical trials, and you'll hear about that today. You mentioned that I work at the Vaccine Research Center, and I think I'll highlight that before COVID, we worked on vaccines like HIV and influenza and Ebola Marburg, but we also worked on what we call emerging pathogens, and that included SARS and MERS. So SARS arose in 2002, MERS arose in 2012, and that work that was done by the scientific community on those viruses really enabled the rapid response that you're seeing to what now we call SARS-CoV-2. So that all started in January. As soon as we knew there was a novel coronavirus arising, vaccinologists around the world looked at the sequence, saw that it was a coronavirus, saw that it was related to SARS, and began to do work on the new virus. And importantly, we know a pretty reasonable amount about coronaviruses. So we know, for example, that the virus has a surface protein that's called a spike protein, and that is how the virus attaches to cells in the human body, lung cells, and that's how it infects. So one of the guiding principles is that if we can teach the body to generate antibodies to the spike protein, as well as other general immune responses, the vaccine has a reasonable chance of blocking infection, and if it does that, is a reasonable chance of preventing disease. So I'll say just a few things to start us off about the types of vaccines that are being developed. So, and sometimes we use the terminology vaccine platforms, and that's to indicate the different types of vaccines and different types of approaches. And these include traditional approaches that people have heard about, and these include more modern approaches. So Starting off with what people, I think, are very comfortable with, protein vaccines like hepatitis B vaccine is a very important part of the repertoire being developed. These are being developed by companies like Sanofi and Novavax, and these vaccines have already moved into phase 1 trials. So there's a lot of advancement there. So at our center, at the Vaccine Research Center, and a number of other places, we're taking somewhat of a less traditional approach. And these approaches involve using either DNA or RNA vaccines. They have the potential advantage that you can make these vaccines very quickly. And so in response to pandemics, we like to use these approaches because we can go from a new virus sequence to a vaccine very quickly. And sometimes this is called synthetic vaccinology because we can synthesize the vaccine directly from the sequence of the virus. One doesn't have to grow the virus, one doesn't have to make the protein. And the idea here is that whether it's DNA or RNA, if that's injected into a muscle, the muscle cells take up the genetic material, the cells make parts of the virus, and the body makes the immune response. So there are several companies doing this, and these include companies like Inovio, Moderna, and Pfizer, but there are many others. And there's what I would call cautious optimism that these types of vaccines will work, but we have to wait really to see the clinical trial data because there aren't licensed vaccines, or there aren't experience with licensed vaccines for these newer types of technologies. And then in a related area, some companies, for example, Janssen or AstraZeneca, are using what we call viral vectors. And viral vectors can be attenuated cold viruses like an adenovirus, where the appropriate genetic material is inserted. And the same principle applies. When that's injected, that virus delivers the...
Larry, I think we might have lost John.
Yeah, John. Yeah.
So he's calling from a government facility, so I'm nervous about that. So Larry, I'm gonna pivot to you and ask you to take the thread that John was talking about, and also talk a little about, you're an HIV guy. You have a tremendous amount of background and experience about HIV. How hard was this for you to pivot from HIV to COVID?
Well, I've been a herpes virus guy. I was an influenza guy. I have been a medical virologist all my life. So, frankly, it wasn't very hard to pivot to COVID. I'll just continue first John's thought about the viral vectors, that they are similar to the DNA vectors, that they deliver the gene into the muscle. We make antibodies, and that's what this is all about. All the vaccines are designed to prevent landing of the virus. They're directed against the spike protein, which is the landing gear. So as far as being able to pivot our organization, which has been organizing clinical trials in the HIV field, both in the United States and globally, it was a pretty easy transition for us. We are experienced in mobilizing clinical trials, the type of clinical trials that are required. Require clinical trial sites and experts throughout the world to coordinate the ability of a vaccine to be shown safely and effectively. What's different in this case is that if you actually look at the speed and the magnitude that we actually want to achieve with respect to trying to vaccinate not just the populace of the United States, but the entire globe, and recognizing that children are also part of this sort of overall strategy. We're really talking about trying to, as rapidly vaccinate as we can, 7 billion people in the world. When you think about vaccine development, it's not just the issue of demonstrating whether a vaccine is effective. It's both manufacturing it and getting it into people's arms. And unfortunately, for many vaccines, that has taken years and years and years. For HPV vaccine, we're still at only 60% or 70% for years, 12 years after it's been discovered. That is not going to get us back to the line that you quoted, back to the normalcy, back to travel where people are safe and the economies and the global economies start working. So part of the system that has to be developed is not just to evaluate the vaccine, but how do you essentially shorten the time to manufacturing and definitely shorten the time to distribution?
So what I'd like to do now is I'd like to ask the two of you, first of all, the diversity of people on this call is tremendous. We have some people from the media, we have some people who are immunologists themselves and scientists, and we have some people from the general public. So we have a great variety of experience and background. So I want to just ask a couple of questions to John first. John, tell us a little bit about the types of vaccines. We hear about recombinant protein-based vaccines. We hear about adenoviral vaccines. We hear about mRNA vaccines. Tell us a little bit about that.
Sure. And for the immunologist in the community, what they'll recognize is that these vaccines actually have different ways that they work, and the types of immune responses that they can generate are different. So from a public health perspective, it's important that we have different types of vaccines because we have different options. Some platforms may work better than others. Some platforms may work better in a particular population, an elderly population. Some may be more easily manufacturable. So if you run the list, proteins have been around for a long time. They can be made at very large scale. We know how to do that. But they may or may not be the best option here. We don't know yet. Some of the newer modalities are very fast, DNA and RNA. We can get them into the clinic very quickly, but we've never really scaled them up all the way. And then in the middle are things like traditional vaccines, like just grow the virus and kill it and make that as an inactivated virus, like polio. So that's also a good option. And until we know more, what we're thinking is, let's take multiple pathways, figure out which of those work. And if 2 or 3 or 4 of them all work, all the better, because in different parts of the world, there may be the capacity to make one more than the other.
And John, that's a really interesting comment. And Larry, it comes back to something you and I were talking about at the Hutch the other day, when you said to me, Tom, it's imperative that we have more than one type of vaccine. For the general public, why not just have one great vaccine and make it for 7 billion people?
Well, there's not one manufacturing plant or a series of manufacturing plants geared for one technology that actually can make anything for 7 billion people. And it takes years, 3, 4, 5 years to actually build a factory and get it certified. So we actually need to use we hope and want to have different platforms so that all the manufacturing facilities for that platform can actually be mustered in order to have a vaccine type. And in addition, some vaccines, like live viral vectors, are not appropriate very often for pregnant women. When it comes to children, we're most used to giving protein vaccines. The nucleic acid vaccines would be really pretty. When it comes to children, we're most used to giving protein vaccines. The nucleic acid vaccines would be really pretty new for children, and what are going to be viewed as the risk-benefit ratios associated with that. So the reality is the best thing for the world, both financially, competition between vaccines and cost, from a safety point of view, to cover the diversity of the experiences of the human, whether you're pregnant or non-pregnant, elderly or not elderly. Different vaccine types are likely to give us different kinds of immune responses.
Okay. And John, in the article, in the science article that you guys wrote, you talked about the fact that you're pretty sure that vaccines will work. You're not 100% sure the vaccines will work. Tell us a little about that certainty. What do we know about, and again, this changes every week or every 2 or 3 days, what we know about the immune response that patients have who've been infected with the virus? Does that provide protection? And this has already come in from one of the people in the public who've asked this, is having had a COVID-19 the same as having a vaccination for it? And do you feel confident that vaccines will work?
So we don't know enough about this disease yet, but we know about other coronaviruses. So there are other coronaviruses that infect people, and these cause common colds. And so one of the ways that we think about it is that the reason that those coronaviruses cause common colds is that there's some pre-existing immunity and they don't progress to more severe disease. If you look at some of the more severe coronaviruses like SARS, which do cause more severe disease, that was a new virus to people. There wasn't any pre-existing immunity. And once one gets infected, there's a suggestion, and there's data that suggests there's a very strong immune response that should protect against reinfection. So we need more epidemiological data to show that. But generally for a virus, especially a virus where the immune system can see it and make a response and clear it, like coronaviruses, generally speaking, that should offer some sense of some ability to protect, although we don't know how long that protection will last.
Yeah. And Larry, I'm going to throw a tougher question to you, which is, timelines. Okay, now you've been working on an HIV vaccine since 1999, and we don't yet have an HIV vaccine. John seems confident that the immune response is going to actually work in this setting. And then we hear questions of timelines. If it's 21 years and we still don't have one in HIV, we know that for Ebola and others, it took several years to get to that point. And yet, we hear from very credible, thoughtful, smart people that we may have a vaccine by the end of the year. I read this morning in one of my cancer journals that one of the manufacturers plans to have 300 million doses available by December. So tell me a little bit about what you think about the timeline, and how should the viewers of this podcast, how should they interpret all the different thinking about timelines that we're hearing?
Well, first, before I answer the question, I want to say that in the vein of misery likes company, John's been working on HIV vaccines with me for the same time.
Okay.
So we're looking for a successful boat to be in together, and we think we have better chances because this genetic mutation in this virus and where the vaccines are directed and it's landing here, we think is an easier scientific target. So we'll then transition to your question about the timelines. Our colleague, our friend, certainly America's Dr. Fauci has been on there telling both of us that, we're supposed to find an answer by January. Now, everything will have to go incredibly perfectly if that's going to happen. Obviously, the first vaccine out of the box, which is this RNA vaccine, is actually, whether it's Pfizer or Moderna, are going to have to work terrifically well to get an answer by January. The higher the efficacy, or the better the efficacy, the shorter the time is needed to show that a vaccine actually shows an effect, as the number of cases occur are much fewer in the vaccine group than they are in the placebo group. So could that potentially happen by January? Yes, it could potentially happen by January. I don't think either John or I are particularly happy with Tony telling everybody that it's there by January. But if everything goes well, that's definitely possible. The trials we're doing are very large, 30,000 persons per trial, and they're overpowered so we can get a rapid answer. So that's one of the strategies we're using. And the trials will require the cooperation of the pharmaceutical industry, the academia, us, as well as the biotech industry, and the NIH.
So John, we hear there are 5 that have been selected, 5 vaccines that have been selected as the chosen ones. I know there are many others that are out there. Can you tell us about those 5 vaccines? And Larry referred to the fact that there's been excellent collaboration between industry and government. Tell us a little bit about that from the government perspective.
Sure. Well, first of all, there aren't 5 vaccines that are selected, so I'd like to clarify that. There are a number of vaccines that are in different stages of development. From the U.S. government perspective, one of the mechanisms that we have is that there's an organization called BARDA, the Biodefense Advanced Research and Development Authority, which allows the government to work with a company and fund vaccine development. And so there are a number of vaccines that have been funded by the BARDA organization, and we can work with them, with Larry's group and my group at the NIH, to help advance those studies. There are other vaccines that are in earlier stages. They may get funding as they're down the road. That could be a few weeks from now, a few months from now. So it's an ongoing, active process of understanding what kind of vaccines are out there, which ones should advance to the clinic. The ones that are most advanced, as Larry was just mentioning, RNA vaccines, because of what I mentioned earlier, they're rather quick to the clinic. They may enter phase 3 trials early this summer, but soon after that will be the viral vector vaccines. And in fact, one of the viral vector vaccines, one that's called Chimp Ad-Ox1, has a long name, that's already in the clinic and started in Great Britain. And then traditional protein vaccines, which could be very important, good vaccines. They just take a little longer. They may come in the early fall. So all those platforms will be in people in early stage over the next few months, and some will start phase 3 in the next month.
And so, John, so what is the media's been carrying there've been a lot of stories about the 5 that have been selected. Can you just clarify them what was either right or wrong about those stories?
I think the way to look at it is when the U.S. government works with a partner, a company, which is called a sponsor, they sponsor the vaccine because they make it, and we partner with them to co-develop it. That information is publicly available, and the ones that are listed right now are companies called Moderna, AstraZeneca, Sanofi, Janssen, and Merck. So those are funded, and we're working with them, and there may be others soon.
Okay, got it. Excellent, excellent. So Larry, as you mentioned, you and John have been working together for years on HIV vaccines. Can we take some comfort in the fact that maybe some of the infrastructure for HIV vaccines could be used to help in COVID? Is that possible?
Well, that's not only possible, that's what's happened. It is this infrastructure that has been built, this collaboration of clinical trial sites tied to a statistical center. The statistical center is at the Hutchinson Center. We have some of the best computational biologists in the world. We certainly have actually the best statistical vaccine scientists in the world, led by Peter Gilbert. And that has been an enormous part of sort of shifting this infrastructure into designing efficacy trials. And more importantly, what we hope to come out with is what we call a correlative protection. If we can show that multiple vaccines work, or at least 2 vaccines work, and we actually can show that there's a biomarker of that working, like a neutralizing antibody or the amount of antibody that binds to the spike protein or the receptor of the virus, then that really allows us to change the way you bridge down to children. That changes the way to get into other kinds of populations, and it actually may allow the protein vaccines, or the ones that go later, to be done through immunogenicity trials. And so the whole world benefits from that. And so a lot of this complex infrastructure that we have developed in HIV, which is by far the most complex virus from an immunology point of view, is being applied to SARS-CoV-2. So it is the laboratories that are associated with this. Some of them are at the Hutch. It is the statistical modeling group. It is the computational biology that knows how to analyze the modeling. It is all the work that we've done in correlative protection that has been essentially pioneered in HIV, that then was extended to dengue and is now being used in COVID-2. It is, we are standing on the 20-year infrastructure that we have built. And I would only say thank goodness that exists, because how else at the moment would we be able to, within 2 months, take 65 sites in the United States, get them mobilized, and are ready to start a vaccination program in July, when frankly, we just started this in March.
Exactly. And so John, so just to summarize, there'll be at least 5, but more is what I'm hearing, large randomized trials from what you guys are saying, maybe 30,000 people in each trial. I mean, it might be smaller or larger depending upon the actual study, but big numbers of patients will be needed to be in these trials. And they'll have a placebo group. Is that true? There'll be a placebo group. And what will the endpoints be in these trials, John?
Well, so Tom, this is a critical point, that there's really no shortcut to an answer here. All the vaccine scientists in the world and the clinical scientists in the world can make predictions or be encouraged by the data. But the only data that matters at the end of the day is if we do a placebo-controlled study and can confidently say the vaccine works. It reduces the risk that somebody gets severe COVID. Right? We may be able to actually look to see if whether you actually get infected at all, but most importantly, one doesn't get sick. And if the vaccine prevents illness, then it works. So the studies are rather complex, because we have to not only vaccinate a large number of people, include a placebo, but when people come into the system with symptoms, if they get diagnosed with, usually now it's a molecular test and nasopharyngeal swab, we have to follow them for some period of time to know, would they get sick because they're in the placebo group versus less so in the vaccine group. So all of that takes some time, and it may vary by vaccine, it may vary by population, may vary over time. So there's a lot of data that has to be generated before we can be confident in a vaccine.
And John, do you worry at all that perhaps with us doing well with social distancing, that the amount of virus in the population might be coming down, that it might make it really hard to see the efficacy of a vaccine?
I think Larry can comment on that better than I, but I think that part of the unfortunate reality is that there is still a lot of transmission in this country.
Okay.
Okay.
Larry, so go ahead.
Well, we're still seeing 22,000 cases of COVID just, in reported new infections per day in the United States. So, so we have plenty of COVID going on, and I think, the reality is I think we're seeing this virus living with us. I think the hope that it's going to disappear, I think is. Very, very unlikely. We still see persistence in microepidemics throughout the country, and I think that is going to occur. And our job is to sort of do the vaccination campaigns where the virus is. Led by Jim Kublin here in our operations group, we are working with HHS and predictive analytics to be able to sort of track where the virus is, to match that with where we would do the vaccination campaigns, and also to make sure that we enroll the populations that are at highest risk. And frankly, this is one of the sort of new worries. Our underserved populations, our Black and Hispanic populations, we are going in as a U.S. government organization, and the events of these last few weeks are going to, affect our ability to establish medical trust, do the kinds of things that we need to do with community outreach. That becomes really a central issue for us to be able to enroll the people who have been most affected by COVID-19. It has shown the health disparities in our country. We are not immune to the societies in which we live in, and the clinical trial infrastructure and how we represent it, how we represent the Hutchinson Center, how we represent the National Institute of Health are all rolled into the issues of us actually pulling off these trials effectively, expeditiously, most importantly, safely, and with the kind of veracity that at the end we know that we have done an incredible good job in finding out whether these vaccines work or don't work and how safe they are.
And Larry and John, that's an incredibly important point about how do we make sure that the populations of patients who are in the vaccination trials reflect the population of the country. And we actually have gotten some questions that say, how do we try to ensure that marginalized communities, people of color, LGBTQ communities, Native, Indigenous peoples, how do we work to be able to offer vaccines? And then if we offer vaccines, how do we deal with this trust issue, Larry? I think you've hit on it well. And John and Larry, I'd like to hear both of your thoughts on how we can get this done, because it seems to me that will be an important challenge for us.
Well, the good news is that we've had a fair amount of experience with that in the HIV field, building trust with communities, recognizing that you have to partner, that the dialogue in community, what community engagement means, listening to the community. It's a two-way street. It is really partnering with community organizations, involving them in the research process, listening to their needs, and trying to create the kind of partnership that allows trust to happen, because altruism and trust is what's required. There is essentially a contract between the clinical trial site and the participant. It's a randomized, double-blind, placebo-controlled trial. We need to make sure that the safety parameters of that are occurred. I have no doubt about that. And frankly, if someone develops COVID, that the medical monitoring in the trial is at the level of the highest standards that anybody could possibly have medically. And I feel confident that we have great investigators. This is what we've been doing. We've been trained to do this in HIV, and we are extrapolating that training to COVID-19, and we feel comfortable that we will not enter into the trial unless we're able to do that.
Yeah, and John, your perspective from the NIAID, how does the National Institutes think of this?
I think, well, we look at it in a very similar way. And in fact, the network that Larry Corey runs with his colleagues, starting with HIV, called it HIV Vaccine Network and HIV Prevention Networks, have really been put together under the organizational leadership of Larry and his colleagues because of the experience they have in doing this with other diseases over the years. One, it's not just a scientific problem. It's not just a clinical operational problem. It's to do a clinical trial like this, where we're actually testing a vaccine, requires a lot of things, and very importantly, the trust of the people that are volunteering. And so we look at it very similarly, and it's an ongoing process as we move towards doing the studies.
Incredibly important. And I want to just think about a different population of people for studies, because obviously this was in your science article. We've heard a little bit about this, and I'd like to actually be curious what you think, John, and then Larry, about these challenge populations. I mean, there's populations of 19 to 29-year-olds who are out there who may be in excellent health. We seem to think that population, they can get sick, but most don't get very, very ill. The challenge population, would that speed things up dramatically? Or, as you point out in your paper, there might be some obstacles there. And comment a little bit about these challenge trials. What are they? And have you seen examples where they've been done well before? Could they help here?
So I think, Tom, you're asking about what we would call a human challenge study.
Human challenge study.
Yes. So those types of studies have been done for diseases like dengue, for influenza, and for others. There's a couple of obstacles here. One is, even for malaria, for example, where people are challenged and we learn about correlates of immunity. In those cases, generally speaking, either we have a treatment for the disease, like malaria. One can give a volunteer malaria to see if a vaccine works. If the person gets infected, they can be readily treated. So we feel like that's an important aspect of it. Or we have such careful control of the virus that we can make the challenge virus attenuated so that we know that the person will be safe. So it takes some time to get those human challenge models in place. And that will be done in parallel, and they can be useful. You can learn about human immunology. But currently, given the timelines we have for the large vaccine trials, it's likely that the vaccine trials that we're talking about here, that Larry's organization will be running, will take place well before we have these human challenge experiments.
Okay, excellent. So before we get to some of the more specific questions that are coming in actually quite briskly from the audience, I just want to finish by asking each of you, if this doesn't work, scientifically, why doesn't it work, Larry? Meaning, we see such promise. We've seen the Moderna vaccine seems to, and I think the J&J also have been shown to have neutralizing antibodies in some of the early patients. If it doesn't work, why doesn't it?
Well, I certainly can't answer that question. I think we have learned out of experience with HIV to design our trials so we can, we're not just vaccinating and bleeding. We have a vast scientific enterprise there that we will get the samples to actually look at the antibody responses and find out why this would not occur. We sort of already know that that some of the neutralizing antibody responses we certainly are seeing in non-human primates are from vaccination are higher than we see in natural infection. There's a very broad range of that, so we don't really know what levels of neutralization will give you protection. So we have to, hope that the experiment works and learn from it to know why it wouldn't be. There are second-generation vaccines that are sort of emerging in the pipeline. They are more diverse in their structures to design to make antibodies to different components of the virus, not just the landing gear. One of the issues would be are the titers higher than we think they are? And we actually need to boost the titers up. It's sort of a little bit of an engineering problem. You can give higher, a better adjuvant. We put in more protein. We're just not quite there. We should then see partial efficacy, 30 or 40% rather than 60, 70, 80%. So we're optimistic and cautiously optimistic that we don't have to answer your question, Tom. But we are geared as scientists to be able to collect the samples and have the scientific wherewithal to figure it out.
And John, if they don't work, why do you think they won't work?
Well, we have experience with vaccines that work and vaccines that don't work. So just, it's interesting from the perspective of the public, we know what vaccines we all get, right? So those are the ones that work readily. We get measles and mumps, rubella, we get polio. But as an example, the most common infectious disease cause of hospitalization in young children is respiratory syncytial virus. We don't have a vaccine for that, despite trying for more than 30 years. So as scientists, we have a sense, well, why is that? Why do we fail there? Why do we succeed in some other cases? Our sense is that for coronaviruses, we're more on the side of the vaccines which work. We know enough about the science to make that happen. But I think what Larry said is sort of the key answers. What often happens is one doesn't hit a home run. You do a study, I think it's likely we'll have some success. We'll induce some kind of protection. It may not be perfect. It may not be the same in all populations. We don't understand why some people get severe pneumonia, some people don't. So there's a lot of unknowns here, and we'll do the right kind of study and we'll build on it. And over time, we'll get better and better, unless we're fortunate right off the bat.
Exactly. Let's turn to a different issue. This is a lot of questions are coming in on this point, and so I want to just address I want to get us thinking about side effects and things that could happen with vaccines that aren't things that we're hoping for. Larry, can you comment on the potential side effects that these vaccines could cause?
Well, the question is really looking at, I think, from a sophisticated person that someone is asking about vaccine-induced enhancement, and in which actually vaccination actually leads to a kind of antibody that doesn't really prevent infection and then a subsequent immune response that actually enhances the disease. And something we've seen in dengue and in John mentioned in respiratory syncytial virus 60 years ago in the first inactivated vaccine that was tried on children, really now essentially almost 60 years ago, that enhanced the pulmonary manifestations when they actually got RSV. I'll only say that each of these vaccines are going through an animal model, and I'll let John's really an expert in this area and let him, talk a little bit more about this. So everything we're doing in animals in preclinically before we get in humans, there's a type of immune response that tends to occur, but yet that doesn't mean it is actually predictive of vaccine-induced enhancement. We actually don't know the really total underlying aspect. And also the studies are overseen by an independent data safety monitoring board, 7, 8 really experienced physicians and statisticians who essentially, once the trial starts, it's under the supervision of the data safety monitoring board. They get briefed every couple of weeks, every 4 weeks about the safety profile of the vaccine, what's going on with the vaccine, and they get all the endpoints. They know whether it's vaccine or placebo. That those data are available to the independent DSMB, and they will monitor this trial as well as the physicians associated with this. We will, we are attuned to this. We only can say that we're moving with speed, but we are not altering safety. We are not. The stages of phase 1 to phase 2 to phase 3, the number of people that you would have in those phases, the safety profile of vaccines to get into an efficacy trial, there are no shortcuts here. The overseeing of the safety is paramount, and the data safety monitoring board will monitor that and is set up to do that in the context of the clinical trial.
Okay. John, what are some of the other side effects besides the enhancement potential? Are there other side effects that we should be thinking about or that you're worried about at all?
Aside from relatively minor local effects where any given vaccine can cause soreness when you deliver it, it can cause sometimes muscle aches, sometimes some fever or what we call myalgia, but those are usually short-lived and what we call self-limited.
Okay. And I guess I'll ask a slightly more complicated or concerning issue, which is, and again, many people, we've probably gotten, a dozen questions on this, which is the anti-vaxxer movement and the concern that the anti-vaxxer groups have that the three elements that they're most concerned about is that the vaccines are rushed, that the government's in cahoots with the pharma industry, and that there are side effects that they're not telling us about. Larry, you spoke about the importance of trust. You also told me when we spoke how important it is to vaccinate as many people as we possibly can to develop immunity as a society. How do we approach the perspective of those who take this position about vaccines in general and about COVID vaccines in particular?
Well, I think one of the issues is to be open and transparent with the data. I think one of the reasons we propose this public-private partnership is to take the whole thing out in the open, to recognize that the NIH and its taxpayers' dollars that are actually being used for the clinical trial, that there is no commercialism in essentially this aspect of defining the safety and the efficacy, that we're going to do it in an open and transparent way, and that it is independent, and it is the National Institute of Health, to which we donate through our taxpayer dollars the major research funds. It is the organization that actually funds the Hutchinson Cancer Center, funds all the universities to do research in our country. So that kind of openness and transparency, when you lift it into that system, we hope helps with respect to showing in a way that independent groups, the Center for Disease Control with their independent panels, the Food and Drug Administration with their independent panels, will define whether a vaccine works or not and how safe it is and what population that is. I think that is a system that has been tried and true. It's been in our countries, I think since the early 50s when the FDA was imposed. It's been emulated in Europe. It's been emulated in every other country with respect to how do you license a vaccine, how do you define whether it's used, and there are multiple bodies that deal with the recommendations as to how it's used. So I think that's in the end, it's the scientific process and the process. I mean, I want John to comment on it. I'll just say different people will approach this in different ways, just like there are different political opinions. And all we can do is present the evidence, do it as transparently as possible, and present it to our country and the citizenry, and hope that they see it the way that the scientific community and the public policy communities also see it.
Yep. John?
Yeah, as Larry said, different people will approach this from very different viewpoints, but I think there's some substantial part of the population that really needs to be reassured. They really just don't fully understand what's going on. I mean, the process, I think if you're a layperson looking at what's happening, is confusing. And so that doesn't generally lend to complete trust. So for the group of people, I think, who are in that bracket, education like this, education from our FDA, from our CDC, from our public health authorities, what Dr. Fauci does all the time, and that kind of reassurance will help a lot. There may be another segment or group of people for which that's a higher bar, but I think we can do what we can do, and I think it will make a big difference.
So a couple of questions before we get to the really critical one of access. One more question for John. How about elderly patients? What specific concerns do you have about, there are many vaccines that are different for people over 60 or 65 than for people under 60 or 65. Do they need different doses of vaccine? Is it just too early for us to even know that now? How do you think we approach elderly populations, John? Or Larry?
Okay, well, John, very good at answering that. I say two things about this. Number one is we are going to be testing the elderly population. There is concern whether it will work as well. There's a very complementary program that we are also running with our colleague Mike Cohn from the University of North Carolina, which again we're the Central Operations Center with his group in North Carolina of giving monoclonal antibodies. And it is the Vaccine Research Center that has pioneered this area in HIV and has also been very active in COVID-2. So vaccines induce the antibody. And if your own immune response, because you're elderly, doesn't do it very well, We actually have these monoclonal antibodies, certainly pioneered in cancer, that we use in cancer therapy all the time, to actually give the antibodies themselves through injection. And antibodies can now last a month, and there are some that can last 3 or 4 or 5 months. So those antibodies are entering clinical trials at the same time we're starting the vaccine trials. And we are actually targeting selectively, persons, the elderly for the monoclonal antibody program preferentially. They give sort of instant immunity, and they're essentially giving the antibodies rather than inducing them with vaccination. So we're concerned about this. The scientific community has developed a strategy for this, and hopefully they will work. And if they work, they're going to be very good sort of corroborators that the vaccination against the landing gear, because all those monoclonal antibodies are to the landing sites of the virus.
Excellent, excellent. John, Garnet Anderson, who's the head of our public health division at the Hutch, asks, do we have any information regarding how long we might think the immunity could last from a vaccine?
Sure. And first, let me apologize for being in and out. There are thunderstorms rolling through, so that's not controllable.
Perfectly understandable.
But what's interesting here is we would normally know that for most vaccines, because we would do phase 2 trials well before we do phase 3. And so you would have time to test the duration of the immune response that the vaccine generates. So here, we just haven't had that amount of time. So we're going to learn that as we go. We're actually going to learn about duration while we're doing the phase 3 trial. And it will also depend on the platform. We have experience with some of these platforms that they induce long-lasting immunity, and I think that will be the case for some of them, but we'll just have to measure it really to see.
So next question is, and again, might be one of the most popular questions we've gotten. This is a public-private partnership. There are 5 companies involved, at least, as John pointed out. The NIAID is firmly behind it. The Hutch and centers around the country like us are eagerly involved. The people who support centers like the Hutch and the peer centers that will be participating are getting behind this. All of us want to make sure access is something we can get to patients, that cost will not prohibit people from being vaccinated. Can both of you give us your perspective on how we can look at that, both in the U.S., meaning is this something you're pretty confident that third payers and Medicare, Medicaid will be eager to jump on and reimburse quickly, and that we'll have access for our population? And then what about outside the U.S., in third world environments? And I know both of you have tremendous experience thinking about that question as well. Larry, if you want to go first.
Well, I want John to go first because I think under Operation Warp, to do the domestic part, and I'll do the international part. So
all right, go ahead, John.
I think under Operation Warp Speed, where John is really a central figure, I think there is, there's been talk about access.
Sure. So this is a central part of the discussion. So as we think about how to test the vaccines and find out if they work, at the same time in parallel, the question is, can we make enough so that we can make it available to large parts of the population? And can it be accessible, and can it be affordable? When the U.S. government is involved, there are a number of agencies there. So BARDA that I mentioned before, the FDA to make sure that the approval process happens, and the Centers for Disease Control, which actually make recommendations on who would get it. But there are built-in provisions already that when the U.S. government is involved in a substantial part of the funding for the effort, as it is here, that in the United States, the vaccine will be available. It will be available to the U.S. government for distribution, and that the affordability part of that can be handled because the vaccine is essentially available to the government as a funder. It gets to be a broader, I think, issue that Larry can talk about as we go internationally, for example.
So I would say that despite what is happening on the outside with the U.S. government and the WHO, maybe we fish who swim deep underneath those waters view that what we're doing is a global program. Our clinical trials program is global. We have incredibly strong sites in sub-Saharan Africa and South America, also in some parts of Asia. And so we plan on using those sites in this vaccination campaign. There are outbreaks in all over Africa, especially in South Africa. People have been reading with Brazil, Peru, Argentina. They all have epidemics, and we feel both our clinical trials program should be viewed internationally. We have communications with the WHO, and our statisticians actually have designed and been part of the WHO committee. We actually know that people in the FDA sit on the WHO committee. So we want to, yes, coronavirus has affected the economies, the travel throughout the world. And we live in a global nation. We are people of the globe. And our plans are to be able to extrapolate all the data that we have in the United States to internationally. And in some areas, we may get more data and in different kinds of populations internationally, which we can back extrapolate to the United States. And that should always be recognized. Certainly in the area of HIV, what we do globally is actually probably more benefit to the United States than we do in HIV to the rest of the world.
And a question, Larry, along those same lines, do we have evidence from other vaccines that there could be racial differences in responses between people in Asia, people in Africa, people in the US of different ethnic groups? Does ethnicity or racial background ever make a difference in how people respond to vaccines?
It's a good question, and I'm not aware that we really have a vaccine that's licensed that crosses racial bounds. It is our immune systems are really geared to be 99% whatever. There are diseases and in which certainly race has different areas of severity, often related to a genetic difference in HLA type or in a genetics of your innate immune system. But highly effective vaccines really have gone beyond race type. Age has been probably a more differentiator. Malnutrition, I mean, it's not the genetics of race. It is the environmental aspects of the human condition with respect to with respect to nutrition, parasitic diseases that affect the immune system, and age. Those things certainly affect vaccination strategies and vaccination efficacy, but not the genetics of race very much.
Okay. John, a question just came in about whether the U.S. defunding of the World Health Organization will hurt this vaccine effort or not. Will we be able to develop these vaccines globally as fast? Were they going to be part of the vaccine development, or is that a different issue?
Right now, the World Health Organization remains intimately involved, working with a number of groups, working with a group called CEPI, which is a vaccine, international vaccine group, working with Gavi for procurement of vaccines. So while, as Larry said, in the pool we swim at our level, that those interactions with the WHO, scientific interactions, public health interactions are going as usual, and it's going to be absolutely critical for the U.S. effort to be linked and coordinated with the international effort through the WHO.
Excellent. And one other question that's just come in about as we head into, and this was brought up actually by the leader of the CDC in some of his comments, as we head into the summer and then the fall, Larry, your thoughts about the interaction between the coming influenza season and COVID? Will the influenza vaccine process affect the vaccination process for this? Where do we stand with that? Will people who are on these vaccine trials be able to get the flu vaccine?
We ask great questions, and we probably will accommodate to the issue of the flu vaccine. We haven't actually dealt with this yet. It does interrupt the issue of developing sort of the side effects as it relates to vaccination. It shouldn't affect any of the immune responses, and so we'll have to sort of accommodate that. And the questionnaire gave me a oops, we haven't really accommodated that in the protocol yet. So thank you for that. The issue of will it affect the clinical trial? Fortunately, we have ways of differentiating between flu and COVID, and many of us, as we move more into flu season, will flu predominate. This year, COVID overtook it enough, and we went into social distancing. So if you look at the Seattle Virology, UW Virology statistics when we went into social distancing, flu just actually rocketed to zero. It is like in all my 40 years of working in virology, I've never seen anything like that. Again, showing what closing schools and social distancing can do to airborne respiratory diseases. But yet you actually saw that in many respects, the tighter we get out of the virus, out of the nose with coronavirus, is about 2 logs higher, actually, than we get out of flu. And so we still saw a lot of transmissibility, and we never saw anything with flu. Now, it wasn't, in my opinion, just because we closed the schools. Kids aren't the only vector of flu. I think that we have a formidable pathogen here in its ability to infect the human being. And it's one reason why I think we're going to live with this virus for a long period of time.
So we're getting toward the end of our time. I'm going to ask both of you to think forward and tell me what you think will be the world of COVID will be like in December of 2021, not December of 2020, but December of 2021, which is 18 months from now. And John, what will we have therapeutically, vaccination-wise, available for people at that point? What will the world, and listen, it's not fair. It's a totally unfair question. I realize that. But there's a lot of people who are wondering what that's going to look like in 18 months.
I think there's a reasonably good chance we'll have effective therapeutics. We have small molecule drugs, we have antibodies. And even if one of them works or some of them work, just think of what a huge difference it would mean to know that if you get sick, there's an intervention that's going to keep you out of the intensive care unit. I think we'll be there. We'll have to see. I think we'll be in a place where we have vaccines. I think the challenge will be how well distributed are they? Are they available to everybody? But with just even a little bit of an optimistic view, I think by that time we'll have vaccines that work pretty well.
Okay, Larry?
I'm going to plan to go. I'm going to be outrageous. I'm going to say I'm going to plan to go skiing in December of 2021, which means A, we're going to have vaccines that are effective. B, I'm going to be vaccinated, and C, there's going to be monoclonal antibodies also. So I think the only way out of the epidemic is through science. But I think we have sort of the beginning to see what the scientific community and the scientific establishment can actually do in a rapid period of time. As they develop vaccines, I think the monoclonals will play a major factor in this. Maybe we'll even have an antiviral emerge by that time. But I think we're starting to see this, the power of the scientific establishment start developing approaches, which I think are the only way out of this epidemic. So, I'm cautiously optimistic because I think this physical distancing is difficult, and we need to hope that science can change it.
Great. Well, I want to thank both of you. Larry, I want to thank you for everything you're doing at the Hutch for our institution and for bringing patients into this network to be able to offer them options in these trials. And John, I want to thank you for everything you're doing at the NIAID from the government perspective and helping to lead this public-private partnership, which is going to be so crucial to us getting there. So I want to thank all the people who joined us today virtually. We're very excited to kick off the Science Says series to dig into more of the research on coronavirus. And we'll also be doing these on cancer down the road. We'll follow up with some more information on future events. I want to wish everyone a wonderful weekend, and thanks for joining us today.